Hi, good morning, everyone. Welcome to the last session of this workshop on knowledge-guided machine learning. To start with this session, so we have today the session focusing on translational biology. So as you all know, in biology, our bioinformatics field, we have tremendous data generated in the past decade. And with the new technology, we have even more data generated every day. So this, so bioinformatics is a big data problem fit into the HDR goals. So especially with the multi-omics data, and we have all different multimodal data generated for each patient. And in addition, with the data, we also, with the long history of biology, and we have generated a lot of knowledge and very well designed knowledge base and ontologies, for example, gene ontology, protein ontology, and disease ontology, and all these prior knowledges can be integrated into the machine learning model, so we can have more precise predictions and with the knowledge guided process. So translational biology is on this path to use the knowledge guided machine learning for better prediction and classification, and to support more personalized medicine for patients and for well-being. So for this session, we have put together 5 lectures from the very well-known researchers and who are in the front of the cutting-edge research. And I hope you will enjoy the talks through the morning and also the early afternoon. And thank you for participating. So I will introduce now the moderator, Kevin James from University of Virginia, to start the session. Thank you.
Great, thank you very much, Aidong. Good afternoon, everyone. My name is Kevin James. I'm in the Department of Biomedical Engineering at the University of Virginia, and I will be the moderator of today's session on translational biology. Aidong, would you be able to put those slides back up? Thank you. First off, if you can go back one slide. As the moderator of the session, I wanted to remind the audience that we will be using Slido to field questions from the group. So I have the Slido app up on another screen here. I'll be monitoring the group for questions. Feel free to put them on there in real time. The link and QR code is on the right-hand side of this slide. And then I'll make sure that we get through as many of the questions as we can during the time we have allotted for each one of the speakers. You can go ahead one slide. We have a nice docket of people from a variety of institutions and also a variety of domains in the overarching umbrella of translational biology. We'll hear about evolution, macromolecular structure and function, as well as semantic types of learning applications, biological applications to cancer study and prediction, as well as more population scale work in epidemiology. Our first speaker is Tandy Warnow, who will give a couple minutes here to get set up with her screen. She's joining us from UIUC, and I'll let her speak to her title, but we can, I think, do the handoff of the screen share to Tandy right now, just to make sure that everything goes smoothly and get an on-time start.
Okay, how's that? Is that visible?
It is. It looks good. And since we're on top of things, I would say you can get started a couple minutes early, that much more time for questions, or that much more flexibility in your opening remarks. It's the benefit of going first.
Thanks. So as Aidong has noted, there's a huge amount of data that's been generated. This is generally genomic sequencing data, among others. And what I'm going to be talking about is how to use the volumes of genomic sequencing data to try to get at reconstructing the tree of life, which is a fundamental model for understanding biology. So this is an example of an evolutionary tree, otherwise known as a phylogeny, showing that human and chimp are more recently have a more recent common ancestor than they do to gorilla or to orangutan. So it's just a tree that describes the evolution of a set of species. So why is evolution important? It's because it gives you a context in which to answer biological questions, evolution that is. Specifically, nothing in biology makes sense except in the light of evolution, a very famous saying that everyone always repeats. Another saying that's a little bit less well known is nothing in evolution makes sense except in the light of phylogeny. So phylogeny is this graphical model, which is why as a computer scientist, we're interested in this reconstruction problem. So when you look at genome scale data, you get something called phylogenomics, which is phylogeny estimation using whole genomes. And so there's this group called the Genome 10K, and there's even bigger groups now that are going out trying to assemble genomes from across all of life. So you get these massive data sets, and you're trying to use those massive data sets to understand how life evolved on Earth. So phylogenetic inference as a computational and statistical problem is fascinating. It has everything in it is NP hard. There's a lot of statistical estimation problems. So you have to do modeling of evolution. You do analysis of the algorithms under probabilistic models, and it's very complex. And I will give some ideas about some of the work that's in this area that is characterized by big data, heterogeneous, large, model misspecification, and everything becomes challenging. And that's why it becomes interesting for us. So this talk I'm going to be giving just a very quick intro to what phylogenetic estimation is about, and then talking about how to do genome scale evolution using a method called Astral, and then how to scale methods for tree estimation to ultra-large data sets using a divide and conquer strategy called tree merge. And then I'm going to go back downwards. Instead of thinking genome scale, I'm going to think about a single gene, because that's the basic unit at which everyone works. First, you get your gene trees, and then you use all that to do the genome scale evolution. So there's a lot of heterogeneity within a single gene in terms of how it evolves. And I'll describe two attempts to address that heterogeneity, but both of them fail. So that's where we get some interesting research challenges and then discussion. Okay, so the pipeline for doing a phylogeny, first you have a lot of data and you go and you annotate the genomes. And one of the things you do when you annotate genomes is decide which regions of the genomes are called orthologs, which is to say they evolved from a common ancestor by substitutions, or which ones evolved by duplications, those are called paralogs, that's different. You get multiple sequence alignments of the different genes. You then construct the gene trees, then you construct the species tree. And then you use the gene trees and the species trees together along with the alignments to do all sorts of post-tree analysis, such as detecting selection, figuring out the dates at which different events occurred, et cetera. So there's this beautiful quote from the National Science Foundation 2002 report that says, resolving the tree of life is unquestionably among the most complex scientific problems facing biology and presents challenges much greater than sequencing the human genome. And I'm hoping to get across some of those challenges to you today. Okay, let's start with an idealized model of sequence evolution. So I want you to imagine that at the root of the tree is let's say a genome, but we're just taking this stretch of the genome of this length. So what you want to think is that the leaves of the tree are species. So like human, chimp, gorilla, orangutan, bonobo, whatever. And what you have is the genome for each of those species. Now you're tracing the evolution of the genome, but just one region. Okay, so the sequences are not changing in length across the tree, but the letters are changing. So those letters are changing because of substitutions. Now that's a visual depiction of that evolution over time, but you need a mathematical model to be able to work with it. And so here's the common way that this evolution is modeled. You assume that the different positions in the sequence, that is to say, this sequence has 7 letters, and so you're going to imagine that it has 7 positions. The different positions or sites are going to evolve down the tree identically and independently. And so all you need to do is model how a single site evolves. And you can just make very simple models with substitution probabilities, and that if you change, and you're currently at the letter A, that's one of the nucleotides, what you change to. And so these are very simple models, and you can make complex models, you can make simpler models, but they all have this property of being IID evolution across the sites. And so with that IID assumption, you can actually do inference. And so you can take sequences that have evolved down a tree and try to reconstruct the tree. It turns out that these models are all time reversible, so you can only get the unrooted tree. So you consider yourself successful if you get the correct unrooted tree. Okay, so phylogeny estimation is therefore posed as a statistical problem. And so you're assuming DNA sequences are generated on some unknown model tree, and you try to infer the tree from the observed sequences that you see only at the leaves. So you can formulate this as like a maximum likelihood problem. That's NP-hard. You can use distance-based approaches. You can use parsimony, which is basically a non-parametric approach. Lots of different methods. Everything that's worth doing because it gives good accuracy is NP-hard. The data sets can become very large. There are phylogenies with 1 million sequences or 10 million sequences. Trying to solve anything well is very challenging. Basically, you can't solve them well. And you have literally years of CPU time for standard methods that don't solve things exactly, even on just moderately sized data sets. So this is very, very computationally intensive. Okay, so how do we quantify error? We have the true tree, which is rooted, and it produces DNA sequences, and then we estimate the tree, which is unrooted, and we just compare the true tree and the inferred tree according to their bipartitions that you get from the edges. So just to make it clear, this edge here splits S1 and S2 from 3, 4, and 5, and that edge is also present here. So that bipartition appears in both trees. That's a true positive. But this bipartition, which is S4 and S5 versus 1, 2, and 3, is not present in the inferred tree. So that's a false negative. And similarly, you have a false positive. So when the trees are bifurcating, you get the same number of each type of error. So this, in this case, a 50% error rate. So the error rate is the percentage of the tree you fail to reconstruct. Okay, lots of different methods, as I said, maximum likelihood, Bayesian, MCMC, distance-based methods, maximum parsimony, lots of different methods. From a statistical viewpoint, we're interested in whether we can prove that a method is statistically consistent. That is to say, as the amount of data goes to infinity, will the error go to 0? Can you prove that the error will go to 0 with probably going to 1 as the amount of data increases? And so the statistical questions amount to, first, is the model tree identifiable? That is to say, every model tree defines a probability distribution on the sequence patterns. Can you determine the model tree from that probability distribution? Next question is, which methods are statistically consistent under the model? A third question is, how much data does the method need to estimate the model correctly with high probability? And then the last question is, what are the computational issues? And we actually know a lot now. This is work over the last, let's say, 20 years, and we have a lot of information about which evolutionary models are identifiable, which methods are consistent, which methods are not consistent. We have some information about sequence length requirements for methods, but the real take-home message is that the models which are identifiable are ones where you constrain the heterogeneity. If you allow the heterogeneity to be too unbounded, then you don't get identifiability in your model, and then therefore you have no chance of having a statistically consistent method. So the models that have been proposed are simple models to keep the model identifiability possible. So you have to remember that we're trying to do science here. We're not trying to do math. And so simplifying the models in order to have this nice mathematical property could cause a problem in our biological discovery, which is what I'm going to get to as we go on. Okay, so back to this, is a method statistically consistent under the model? There are statistically consistent methods such as maximum likelihood solved exactly. But how much data do you need? Maybe you need genome scale data. So, oh good, we have genome scale data now. So let's take genome scale data. Guess what? Got a problem. There's this problem that as you go across the genomes, the trees change. And so, for example, here you see human and chimp are siblings. That's what you expect. Over here, human and gorilla are siblings. You don't expect that. And what's going on is that human and gorilla are not siblings in the species tree, but for this gene, they are. So gene trees can be different from species trees. And there's lots of reasons for this. The dominant one for many people is something called incomplete lineage sorting. And what I mean by for many people is that some people would say that this is the major cause for gene tree discordance. Other people would say gene duplication and loss is the major cause. But really, incomplete lineage sorting is at least one of the top two. So incomplete lineage sorting is a population level process that has a nice mathematical model. The nice thing about having a nice mathematical model is you can prove theorems, and therefore you can get identifiability and statistical consistency. And that's what we're going to do next. But also, it occurs all over the place. This is a study that was published in Nature about 1,000 plant transcriptomes, and lots of gene tree heterogeneity consistent with incomplete lineage sorting. Same thing for the avian phylogeny published in Science. Massive gene tree heterogeneity consistent with incomplete lineage sorting. So we've got this challenge that we have to address this because it happens in biology. Under the gene evolution model, the species tree generates gene trees that can be different from each other, but it's under a mathematical model called the multi-species coalescent. Then each gene tree evolves sequences, and we talked about the gene sequence evolution already. So if you want to take this hierarchical model and figure out the species tree, you only get to look at the sequence data. So how do you do it? Well, you can reverse the process. You can use the sequences to infer gene trees, and then you can take the gene trees and infer the species trees. It's a two-step process. And there's this nice mathematical theorem that says under the multi-species coalescent, the most frequently observed quartet tree is the species tree, with probability going to 1 as the number of gene trees goes to infinity. And that gives you a method. It gives you a proof of identifiability and a statistically consistent method, which is to say, just look at all your gene trees, figure out the quartet trees, and then combine the quartet trees. And so that's a method that was essentially solved in Astral. It's an NP-hard optimization problem, but if you solve it exactly, you have a statistically consistent method. And so what Astral does is it solves it exactly within a constrained search space. And by constraining the search space, you maintain statistical consistency and you make it a polynomial time algorithm. So Astral has really good accuracy. This is the y-axis is error. The x-axis is the number of species, and it's a comparison between Astral in green to something called MPS in red. And as you increase the number of species, the error rate for MPS goes up. The error rate for Astral goes up very slowly. So Astral is just superior for large numbers of species. Okay, Astral's used all over the place. It's really the dominant method now. This is work by my former student, Siavash Mirarab. And there are pros and cons. The good is it's statistically consistent, it's extremely fast, it's much faster than doing a standard concatenation analysis where you just take all of the gene sequence alignments and put them together and then just run something like maximum likelihood. Sometimes concatenation is more accurate, Astral is often more accurate, but the bad thing is that it can fail to complete. So it fails to complete on large data sets that have a lot of heterogeneity. And so my former student, she just graduated, Erin Malloy, came up with this beautiful divide and conquer routine where you take your set of species and all the genes and you divide the species into subsets. You get species trees on the subsets and then you merge them together. And merging them together using Astral to get species trees on the subsets, and then merging the species trees on the subsets together using the technique that she developed called tree merge. So what's nice here is that if you look at what happens when you run Astral by itself or Astral within this divide and conquer strategy, the tree error doesn't really change. This is just comparison of Astral by itself to Astral inside the divide and conquer. It's not really changing, but the running time is decreasing dramatically. So tree merge has nice theoretical properties. It's polynomial time, it's statistically consistent, and empirically it improves the running time and it doesn't sacrifice accuracy. Okay, so we made an advance in terms of scalability. We made an advance in terms of large numbers of data sets, and it can be used in any context. So the summary is, so far we're doing really well. We're able to actually really scale. The problem is Astral assumes your input is true gene trees. This is not the case. You don't get true gene trees. First of all, gene tree estimation is an NP-hard problem for all the good ways of doing it. Secondly, the model assumptions, as I said, are simplistic. They're not realistic. And if you have unrealistic model assumptions, you get poor accuracy. So how do we address this? Two basic approaches. One is just have more complex models. If you have more complex models, you can actually do better estimation, presumably, right? You can actually try to get a very realistic biological model and do inference under the biological model. The problem with that is that you actually have overfitting. So you actually don't get an improvement, plus the running time goes up. So that leads to the second approach, which is deep neural nets. It's a natural. Deep neural nets give you the potential to train under very complex models. In other words, train your data, train your models on data that are simulated under very complex models, and then you can do inference under those complex models. Now, there are two approaches that have been proposed. They both have the same flavor. They basically train the deep neural nets on simulated data to estimate quartet trees, which means four-leaf trees. They take the four-leaf trees and then they combine them together into a tree on the larger data set. The problem is that these results actually haven't been very satisfactory. Even on data simulated under the same model on which they're trained, they're actually not better than standard methods. And on biological data, it's also not encouraging. And I'm just going to show you some results here. This is our way of writing table 2 from Zhu et al., which was just published in Molecular Biology and Evolution. This is tree error rates. So the three different deep neural nets: neighbor joining, which is a distance-based method; parsimony, maximum parsimony, which is non-parametric optimization, NP-hard; RAxML, which is a maximum likelihood method, very popular; PhiML, another maximum likelihood method, very popular; and then Mr. Bayes, which is a Bayesian MCMC under 5 different biological data sets. And what I've done is for each of these data sets, I've reported the error rate, which is the fraction of the tree that has failed to be recovered. Each of these has these are just quartet trees. So the fraction of the quartet trees that are incorrect. The best across all of these methods is typically neighbor joining or parsimony, not the deep neural nets. The deep neural nets are not as good as simple methods, and even the maximum likelihood methods are not as good as simple methods. So what are we learning here? The deep neural nets are not that good. The non-parametric methods are better. Now, biological truth is not absolutely known for sure on any biological data set, so it's not 100% clear that these error rates should be taken exactly as God-given. But deep neural nets for tree reconstruction don't seem to be as good as standard methods. And the question is why? I think it could be that we actually have models. So we have models of sequence evolution, and even if they're not good, they actually are phylogenetic. And maybe these models that we're working with have the potential to give better trees than deep neural nets, which are not actually phylogenetic models. So another approach for using deep neural nets that has been proposed in a recent paper is to use it to inform method selection. So to say, for this data, we would recommend you use parsimony. For that data, we would recommend you use maximum likelihood. In other words, instead of saying, let's just use the deep neural net to do the construction, let's use it to inform method choice. Another possibility is to inform model selection. Another possibility is to do something where we don't yet really have good models, for example, phylogenetic networks. So I guess this is the topic of this seminar, is about using biological knowledge to inform machine learning. And what I'm showing here is right now it needs more biological knowledge to become useful. And using these things without that biological knowledge is not yet working. Alternatively, use things that are actually potentially more closely connected to the kinds of data we're working with, such as hidden Markov models, which are really well designed for sequence evolution. Okay, this is the literature that I talked about. The first 3 papers have to do with deep neural nets and phylogeny estimation, the paper for Astral, the paper for tree merge, and then 2 papers that are actually about model misclassification and the evidence that is present that, in fact, there are substantial model violations in biological data, and therefore calling for new approaches. And with that, I hope I've convinced you that resolving the tree of life really is challenging. This is a paper from 2002. It's much harder than we thought, even in 2002. It's not just data set size. It's really a very complex statistical estimation problem that needs new ideas. Okay, and so Siavash Mirarab, who did Astral, and Erin Malloy, who did tree merge, are responsible for the advances. And I'd like to thank NSF for the support I've gotten, and the NSF Graduate Fellowship to Erin Malloy. And this work was done on supercomputers, and Blue Waters was, of course, supported by NSF. Thank you very much.
Terrific. Thanks very much, Tandy. We do have a couple of questions on Slido. I'll start with one from Vipin. I think you already addressed some of the ways. You had a question about how biological knowledge could help. I think you already tackled that. There was a question of the seriousness of false positives or negatives with regard to tree construction. And if I could kind of add on an addendum to that, I was interested to know if there were any historical examples of bad trees that then subsequently got improved with better algorithms that led to new insight about the evolutionary paths of different species, organisms.
So you've asked me, I think, 3 questions. Let me just repeat them. One of them is false negatives versus false positives. Another was any evidence of bad trees that have been overturned with new methods. And was there a third question, or were those the 2 questions?
I think those were the two from me and from Vipin.
Okay, so the first one, almost all estimated trees are fully resolved. And when you have fully resolved estimated trees and fully resolved model trees, then there's always the same number of false negatives and false positives. So one approach that people take is to not always return fully resolved trees so that they can avoid false positives. So that is of interest. And a natural way to do that is to do statistical support. And then you can identify how much support you have for every edge. And then you can collapse the edges that you have low confidence in. So there is a desire to reduce the false positive rate. And there are techniques for that. There's very little way to reduce the false negative rate as a post-analysis thing. Now, with respect to trees being overturned, many, many, many examples. I mean, some of the overturning of trees came about because of the initial use of morphology instead of sequence data. And as people moved from morphology to sequence data, things changed. Things are also changing by taking heterogeneity across the genome into account. So as you actually model the heterogeneity across the genome and you use methods that use that heterogeneity to do inference, you can actually change the trees. So there's an improvement through using genome scale data, because you need genome scale data to do species trees. There's an improvement in method choice by having methods that are addressing heterogeneity. And then there's just changes in terms of data set size. As you take more and more sequence data, you have densely sampled phylogenies, and those have better accuracy. So it's all pushing towards methods that can scale to very large data sets, methods that can work well under high levels of heterogeneity. And the challenge, though, is it's not straightforward how to do that. You don't necessarily want to have complex models. But yes, lots of examples where trees are being overturned by new methods and by new data.
Great. We have one question from the group, which I think segues very nicely from your remarks on heterogeneity. And the question relates to how the models handle, again, with maybe a clarification on what is the full scope of heterogeneity. How does it handle errors, for example, like next generation sequencing errors, and then noise, in particular, the diversity of organisms within a species, right? Polymorphisms or other types of things, just variants in the population with regard to real species-specific differences.
Okay, so in terms of errors in the data, so the sequencing reads have errors in general, and people have been concerned about what happens when you're assembling genomes and there's errors in your assembled genomes. It doesn't look like errors in the assembled genomes have resulted in errors in tree topologies because they're low enough. One of the interesting things is that the tree topology, which is to say just the graphical model without any annotations, without branch lengths, that seems to be pretty robust as long as you have a very good multiple sequence alignment and a very good tree estimation method. It can be pretty robust to that. What's not clear is how a lot of the numeric parameters stand up to errors in your input. What's not clear is how the numeric parameters, which is like the branch length, so dates at the internal nodes, detection of selection, all sorts of things, how those things stand up to errors. So I think that's a direction for research. The other question had to do with like perhaps anomalies in your input. Like, was that like the second part was like when you have heterogeneity in your species?
Yeah, exactly. Polymorphic variants or other things that are specific to the individual who was sequenced.
Yeah, so there's two aspects to that. One is that sequences don't evolve just with substitutions. They also evolve with insertions and deletions and duplications. And most methods are not designed to allow data with duplications or data with rearrangements. And so there's just this general issue if you have a lot of data and then you just narrow it down. And so that's one issue that I would say at a very high scale, it's just not solved. The question of like which individuals you're picking within your species to represent your species, and does that impact your species phylogeny? Yes. There is work that suggests you really want to take lots of individuals for each species to get the best results. On the other hand, it's a trade-off. You could add more species instead of adding more individuals. And so there's always this benefit from having more data, but you have monetary issues and just data sampling and collection issues. So it can impact things, and the more data in some sense, the better. One of the things I see all the time is that a lot of people will say, don't use that kind of data, you're going to get bad results. And what they don't realize is it's not a question of the data, it's a question of the methods. So a lot of the time people say, don't use data that evolves too quickly because you can't reconstruct the tree. But in fact, you just need better methods. So there's a lot of benefit for computer scientists to developing better methods. I hope I answered the questions generally.
Yes, great. I think very thoroughly. All right, thank you very much, Tandy, for the talk. Appreciate it. We're going to try to keep just that couple minutes ahead of schedule so that we have a little bit of overflow as needed. Our next speaker is Amarda Shehu from George Mason University. Amarda, if you want to get your slides set up.
Yes, let me see if you can see them. Are they visible?
Yes, they are visible and I can hear you fine. So the floor is yours.
Perfect, thank you. So I am going to continue on what Tandy started, and I'm glad that she mentioned at some point going all the way down to a single gene, because I'm gonna take us a little bit more down at the gene product, and I'm going to talk about macromolecular structure and how that impacts our ability to understand function. So I'm gonna get us started by thinking about sort of an organizing principle of a lot of research in computational structural biology, where we worry about how things look, what forms, what shapes they take. And I want to credit Sir Turing, who in a seminal paper in 1952 really posed the question of how the form of matter determine function. In biology, we see an instantiation of this question when we worry about how DNA gives us information about cellular function, and how the gene products, the proteins, give us information about the biological function or the biological activities of proteins. But if we want to understand function, almost certainly we have to sort of produce another question, and that's the question of, all right, if the journey to function goes through understanding and modeling form, how do I represent form, and how much information and how much detail should I have in my representation of form? Unfortunately, another giant of science, Aristotle, is not that much help, because he basically says that, all right, try to work with an approximation of the truth if representing all the level of detail in form is computationally infeasible, and that is almost certainly the case in biology. So trying to represent form, this is something that my group has thought about a lot over the years, and it's a challenging question because two issues arise. One is representational detail and the other is scale. So when we talk about representational detail, we're basically saying, does just knowing the building units in a molecule, whether that's DNA or protein or RNA, is that enough to understand what that molecule performs in the cell? Or do I also need to worry about the arrangement of these units with regards to one another? Do I need to go to 2 dimensions or 3 dimensions? And then the matter of scale says, hold on, are you really simplifying the question? Can you get really that information from looking at one molecule in isolation? Or do you need to consider how the molecules come together? Do you need to go all the way up to the cell, the tissue, and the organism? So these are challenging questions. And I will try to organize sort of our journey through trying to answer these questions or instantiations of these questions in sort of these 3 threads. I'll give an example where you can get a lot of information about function. You can infer function just by looking at the building units. not going all the way to 3 dimensions. And then I'll say, okay, this is not enough when we go to proteins. So I will try to figure out how could we represent structure now in order to get information about function. And then I'll add another complication and talk about molecules that are inherently plastic, dynamic, that switch between structures in order to perform the biological activity. And we cannot ignore this if we want to understand their function or dysfunction, specifically in pathologies that are due to proteins. So we've done a lot of work in this first thread, how to go from sequence to function. And I just want to give an example of how biological knowledge can get you to very powerful models. We have studied many different, what I would call recognition problems that ultimately end up being sort of classic classification problems. What I'm showing here in this figure is a promoter region. It used to be a very sort of classic and very appealing problem for machine learning researchers to annotate, to identify promoter regions in DNA. And you could say that in 2012 up to 2014, that it was a subfield that was saturated because a lot of models had been put out there and they were pretty decent in their performance, but their performance had peaked. So we got interested in this question by a collaborator who used to be at Maryland College Park. And our first inclination in the lab was to really understand the biology, to understand that the reason that this particular region in DNA is a promoter that sort of starts that signaling that ends with transcription is because a specific protein, a core RNA polymerase, is able to identify the hidden signal in this region and to bind to it and then start a whole series of events. So we understood that function really encodes implicit constraints, even if we only consider a linear representation, basically only the basis and how they link up with one another in one dimension. We realized that this protein, because it's a 3-dimensional object, when it comes and it binds a region and recognizes it as a promoter region, it's basically recognizing subsequences or motifs that are far away in sequence from one another. So it's imposing non-local or distal constraints, and we wanted to capture them as features. So we had this inspiration that, okay, we can represent these biological signatures if we construct complex features, some of them, for instance, demanding the presence of 2 motifs in different positions. We call these correlational features, in addition to other, the more useful, the more common, I should say, features that everybody was putting together at the time, mainly compositional and positional features. So our idea was to really have sort of a representation of a promoter region in terms of a bag of biological signatures in it, these features that would be able to discriminate it from other non-promoter regions. So we would have a very interpretable answer to how is it that this sequence encodes, being a promoter, encodes function. So we used a lot of ideas from predicate logic, from grammar, in order to sort of build these richer representations of both local and non-local constraints. And we could really construct very, very complex, very rich features by Boolean combinations over basic building blocks. We could have ands, ors, nots on top of sort of other compositions. We used our ideas from how do you explore a high-dimensional space in order to deal with the possible issue of exponential explosion of your feature space by using genetic programming, sort of a biased sampling of the feature space, biased by a surrogate fitness function. And we were able basically to put together a full framework, which we call the effect framework. I can't tell you now what all the letters stand for, but it was something about features and evolutionary computation at the time. And the framework really outperformed many of the methods at the time. It produced features with very high information gain. And more interestingly for us, it gave very interpretable features. If you see here on the right, you can see what the feature is, and a biologist can read off it and say, okay, so it means either a motif at this or a motif at this, and so on. We went and we scoured PubMed literature for the presence of the features that we had found in biological literature. And indeed, we found that many of our features were known. They had been named by biologists. So we were able to replicate existing knowledge, and sort of in a more tantalizing aspect, we were all able to also produce features that were not published in or documented in literature at the time. And sort of this got us to thinking that, hey, maybe we were also advancing knowledge. But all these became because we were able to marry some pretty, I would say, standard methods in machine learning with our information or our knowledge of what it means to be functional, of the underlying biology. We then use some of these ideas in order to sort of even make some improvements in machine learning in order to evolve features and kernels. And as Tandy mentioned, nowadays there's also the sort of this drive to what would a deep neural network do. I resisted it in the lab for some time because I'm always driven to understand the deeper sort of the deeper biology and the sort of the deeper question, but I had students who were really itching to put together a deep neural network in order to drive at some of these same questions when you were where you are able to infer function from sequence. So I had a student at the time who was able to put a rather simple, I would say, a deep neural network with convolutional layers and recurrent layers. The recurrent layers would help us to handle arbitrary length peptides in this case. So we became interested in additional recognition problems, and the specific one that I'm showing here is the problem of recognizing antimicrobial activity from given peptide sequences, so very, very small proteins. So in these ones, we're not considering what form they take in 3 dimensions, just the sequence of amino acids that compose the peptide. And to my dismay, I should say, but to the happiness of my student who we then published this paper, we found that the deep neural network really outperformed all the other machine learning models that were published on antimicrobial activity recognition. But what we sacrificed was interpretability, because we could not very easily go and have this very explicit interpretable bag of the features of the biological signals to say, okay, here, this is what it means to be antimicrobial. So we could get the performance, but really not the deeper biological reason for antimicrobial activity. If we keep going at this question of how much do you need in order to infer function, the answer really depends on the problem at hand. If you go to proteins, for instance, here, almost certainly you will clash, against the wall of structure, of 3-dimensional structure. And it's important because I'm going to show you, so these red threads here are 2 3-dimensional structures, 2 different proteins. These proteins, if we only compare their sequence of amino acids, the building units that compose them, they have very low identity. So a very small percentage of amino acids are identical. But they are functionally similar. Their activities are very similar. And you can see just from these pictures and recognize, oh, the structures look similar too. Yes, so it's very often the case in proteins that sequence is not enough. You need to consider the 3-dimensional organization of the building units, because that can tell you more information about function. So if I put it in terms of evolution, really function places more of a constraint on the 3-dimensional structure of a protein rather than on its sequence. The sequence changes with much more frequency. So now the question becomes, if I'm looking at structure as the form from which to infer function, how do I represent structure? This is a challenging question. And the key insight that we had at the time was, all right, maybe I need to add some spatial information to the building blocks. My idea of motifs needs to change. It needs to consider 3-dimensional information. But we need really succinct representations. They need to be informative to help us answer the problem at hand, but I shouldn't end up with these bloated representations that then will make the problem infeasible. So we had some insight from biology that even though 3-dimensional structures of proteins are very rich and very diverse, they seem to be using sort of a limited library of dictionary, if you want, of short spatial fragments. So when we consider space, we call those motifs fragments now. And so our insight was to, all right, go over known protein structures, compile the different fragments that we see in known protein structures, and then use these fragments in order to represent a protein structure. We used analogies with text mining, basically looking at a protein structure as a document and looking at the fragments as the words, and then using LDA, latent directly allocation, in order to obtain topics distributions over the fragments and end up with a much reduced, much lower dimensional topic-based representation of protein structures. And we showed that this topic-based representation was informative. It helped us to sort of do some inference tasks, predict superfamilies in this case, even in superfamilies that had a very high imbalance, very few protein structures known. And more interestingly, when we started to dig into what do these topics tell us, we found out that, all right, these topics, some of them were present in only few superfamilies, whereas others were present in a few more. So one thing I want to mention is that when you sometimes want to make analogies with other sort of subdomains in machine learning, other challenges pop up. For instance, in text mining, when you obtain topics, you can sort of look at the distribution of words and you can say, oh, this topic captures spores, or this topic captures politics. But when it comes to proteins, you don't have, you know, that lexicon. You cannot explain this topic similarly. So you need to dig a little bit deeper and use other tools at your disposal in order to give meaning to topics. We had a little bit of detour along the way, I would say, when we started to think about function and how to get it from sequence or from structured data. We realized that we could have a lot of data, as Tandy mentioned. So how do you do machine learning at scale, especially for when we were dealing with sequences from DNA, promoter versus non-promoter sequences? We had millions and more sequences. So we had a little bit of detour and put together in the past few years, this was before, I think it was before XGBoost and AdaBoost, we put together what we called a parallel spatial boosting machine learner, where we used ideas on how to distribute the training data over toroidal grids and how to distribute the learning, basically, to have many learners learn locally, but exchange samples with neighboring learners and evolve the training data basically towards the most difficult instances that together sort of patch that decision boundary. And we were able to also publish some theoretical results and get more insight into why PSBML performed as well as it did. I'm not able to tell you about a lot of other problems that end up being, how do I infer information from three-dimensional structure, but in structural biology, they have sort of specific names. So I'm just mentioning them for people that work in this subdomain: model quality assessment, decoy selection. And in this subdomain, we also did some work on manifold learning and more generally in representation learning via I want to say things such as autoencoders and variational autoencoders. So let me try to get to the part of the talk that is of more interest to me, the more sort of recent work in the lab, which considers this additional complication that many macromolecules, proteins, and even RNA, they're not frozen, they're not static, they are always oscillating, they're always fluctuating. So hopefully this animation shows. On the top left here, I'm showing a sheet of cardiomyocytes. These are heart cells that are grown from a collaborator at UPenn. This was a very recent paper that came out where you can see basically the heart beating. And we could determine sort of abnormalities to the amplitude and the frequency of the heart beating. We could trace them all the way down to mutations in a specific protein that was due to binding another one, was sort of the main culprit for several cardiomyopathies. On the bottom right here, I'm showing you a single molecule view. The bottom right is a very important signaling protein that signals cells when to grow and when to stop growing. And what we have understood for a long time about this protein is that it sort of exchanges between two main structures, an on and an off structure. And in one of them, it's basically starting a cascade of signaling events that tells the cell, yeah, you can grow. And in the other, when it's off, it stops the signaling cascade. So the cell stops growing. And many mutations in this protein basically disrupt this mechanism and show up in various cancers. So a key driver in the laboratory is not only to exploit form in order to answer questions about function or dysfunction, but really to go further and to exploit these dynamics, changes to form, to be very cognizant of that. And even though most of the work that I'm showing here today is specifically on biology, a lot of the ideas pertain and allow us to answer, have allowed my lab to answer interesting questions about other complex modular dynamic systems such as networks and bridges and so on. So it's very satisfying to go from one domain to another. So all these structures between which proteins, let's say, fluctuate or other molecules, they have to satisfy constraints. So it's a very interesting question to throw to students to say, okay, can you put together a generative model that can predict basically the different forms, the different structures that satisfy certain constraints, functional constraints or energetic constraints, and to be able to answer specific sub-questions in structural biology, such as structure transitions. Tell me what is the specific series of structures that takes my protein from on to off. I want to see the movie. I don't want to see just the beginning and the end. I want to see the frames in the middle. Or to go even further and say, look, I don't have much information about this protein, but can you tell me about its whole configuration space? Can you tell me about the structures that it accesses? I'm starting from scratch. And when you start from scratch, this is a problem known as protein tertiary structure prediction. We've done a lot of work in this one via classic stochastic search and optimization algorithms. And more recently, we are going in sort of the direction of variational autoencoders and adversarial networks. The reason I have highlighted them in blue is because they don't do as well as our best methods. And you can sort of keep using ideas from sort of generative models in order to map, for instance, what is the landscape, what is the space of all small drug-like molecules. And here we've made actually some headways. It seems that a deep learning can allow us to really generate sort of more credible distributions of small molecules than distributions of larger molecules, such as proteins of a few hundred amino acids. So here, for instance, is a very short summary. This is very recent work. Some of it appeared at KDD this year and at ACNBCB in a few weeks. And this one basically is showing the schematic of the graph generative network that we put together, which was able to generate small molecules that you see here on the top left. The numbers below show you how much drug-like they are. This is a measure that you can actually, this is a metric you can measure. And so here is basically a snapshot of the different molecules that this network, when trained over available data, was able to generate. These networks don't do so well when you now want to expand beyond, a few atoms to thousands of atoms and capture these spaghetti-like colorful structures that you see here. Here we have made better headway with other stochastic search algorithms, but even these ones have to use information in order to map, the rich landscape, such as this terrain that I'm showing to you here. This is an illustration that captures all the different structures of a protein. So we had a little bit of insight by understanding a little bit more about the energy landscape view of protein folding and protein binding by realizing that when a protein, even if when we only know information about it in terms of it switching between two structures, that switching is actually a pathway in a terrain, in a landscape. Every point in the landscape is a different structure, and the height of the terrain tells us about the energetic, the ability of the protein to access, how hard it is to access that structure. So how much time will it take the protein to access that structure at equilibrium, at physiological conditions. So what we were able to do was using known structures, for instance, even of different versions of a protein, we were able to see that these were points in a landscape, and we could exploit this information in order to map, to find the variables, to sort of map the manifold and to conduct the search in the right space. Here is, I'm not going to go in detail over this method, but it is not a deep learning. It is actually an evolutionary algorithm that takes the sort of the variables that you have found, the variables of intrinsic motion among known protein structures, and populates the space with even more structures in order to obtain hundreds of thousands of them. Each single point in this picture corresponds to a structure. I've reduced it over the two principal components. The black dots are known experimental structures, and all the other ones are new structures generated by the algorithm. So I have really a global view of the landscape of this is a specific protein known as calmodulin, and I can even map. I can put a graph representation of this landscape and answer very interesting questions about, oh, how much time does it take to switch from one dot to another dot, from one structure to another structure. And here on the right, I'm showing to you sort of a schematic representation of all the different pathways that we were able to extract from the landscape of how calmodulin switches between known experimental structures. And this view was very interesting because it allowed us to reconcile seemingly contradictory wet laboratory findings and tell us, here is how calmodulin is able to shed calcium and bind protein, either first by doing it in a serial manner or in a gradual manner. So there we were able to reconcile what used to be sort of two different views in biological literature at the time. Here is the first ever landscape, the first ever view of the structure space of RAS, that protein that I was telling you is so important in many human cancers. We were able to take all known biological literature and sort of map it on our landscape and figure out, oh, this is where this allosteric switch happens, because we had for the first time a global view of the structure space accessible by RAS. And more interestingly, we were able to use the same method to map the space not only of the healthy form of RAS, but also of other variants, forms of RAS where there have been mutations. And we were able to see, for instance, that the space, the landscape of oncogenic variants of RAS, they were very similar. They had an interesting mechanism of making the off state more and more inaccessible, whereas other forms of RAS that were causing disorders but not really oncogenic had a very different mechanism. We pursued this even further and we extracted descriptors from these landscapes, and we were able then in training over known biochemical measurements to tell in great detail what specific mechanisms were impacted by the mutation. So this was sort of our first very successful story of predicting the phenotypical impact of mutations through an algorithm that was able to generate more structures and then to infer the landscape and extract important information from it. So I hope to have made a little bit of sense. This was work that has been funded in part by NSF and many other private awards. And the work that I highlighted is done with several collaborators at my institutions and in other places. You can find more information at our website, or you can email me directly with any questions or follow-up. Thank you. Kevin, you are muted.
Thank you, Amarda, for the presentation. We have a couple of minutes. I want to get to two questions, if you can answer them concisely. The first one I think might be short. Is the question is, are the structures that you are learning smooth functions, or do they have considerable non-smoothness? It seems to be the latter from the graphics.
Can you say the latter part? So the structures are not functions. The structures that we have, so now it depends how you represent them, but in sort of the most straightforward representation, the structures we have specify the Cartesian coordinates of each of the atoms that are bonded to one another and that are the units of the structures.
I think that the question stems to the structural transitions that you were modeling.
The structural transitions. No, the structural transitions are not function. Again, so I made this analogy with movies. They are a series of structures that tell us sort of the order in which you have to go through in order to go from, say, your source A to your destination B.
Thanks. Okay, and then the one additional question is, I'm going to just read this one verbatim. Could you expand more on how you assign meaning from topics as defined by LDA? I've experienced it being difficult with text analysis, but curious how that translates.
Yes, that is we were having a hard time. Let me try to go to that slide. I apologize for the nausea this may be causing. So I said in passing that it's generally hard because you don't know. It's not that I can look at the distribution of fragments and say, oh, you know, the word sports appears many more times over others. So I'm going to call this mainly a sports. This document covers mainly the topic sports. So in here, what we did is what I'm showing is really a heat map. So I'm showing basically for, let's say, for a specific topic. And again, you see, I have not labeled the topics. They would be here on the columns. Let's say topic from 1 to A. They are nameless because we don't have names from them. But each topic is a distribution of fragments. So we would see basically where is the topic more prevalent across the different superfamilies. And here, this is basically heat map. In red means more prevalent. In blue is less prevalent. I don't think the contrast is showing very well. It's coming across very well. But this is a way that we could sort of infer meaning to topics and say, you know, topic 1 predominantly is more prevalent on the PILU binding superfamily and the EF hand superfamily, and not so much on the others. It's sort of an attempt to interpret the topics. It's not, I would say, the end all be all, but it is sort of a reasonable way of trying to understand or to link a topic if you want to function.
Right, thank you very much again, Amarda. Our next speaker is Cathy Wu from the University of Delaware. So Cathy, if you want to get your slides set up. All right, floor is yours.
Okay. All right, great. Let me go to the presentation mode. Okay, well, wonderful. Thank you, Aidong and Kevin, for organizing this session. I'm really extremely delighted to have this opportunity to share with all of you about our work in the area of integrative text mining and semantic computing, in order for us to really look into data-driven biomedical knowledge discovery. And I would like to, at the end, to comment on how this may help contribute to the knowledge-guided machine learning. So just a quick overview for my talk. So first of all, I would like to introduce our collaborative research in the areas of literature mining and knowledge integration. So this includes the Uniport knowledge base as a research infrastructure, and our research in natural language processing and text mining, and also knowledge integration for construction of biological networks. Then I will go to the second topic about semantic computing framework, and discuss the scientific use cases that we have developed in constructing gene disease drug networks for diseases from Alzheimer's to COVID-19. And then finally discuss the relevance of the framework to the knowledge-guided machine learning for team science and certainly translational biology. So for our research in translational biology, for understanding of genotypic to phenotypic relationships, it really requires knowledge about proteins, about protein function in phenotypes and disease biology. And we heard beautifully from Amarda's presentation that how we can really study the protein sequence and structure relationship to better understand the function. So as part of the Uniport consortium, our group at the Protein Information Resource, along with our partners at European Bioinformatics Institute at UK and Swiss Institute of Bioinformatics in Switzerland, we have developed a Uniport knowledge base as a central international resource to put together comprehensive protein sequence and function information for all organisms from human to microbes. And so in the process of putting together the resource, our goal really is to connect with the vast resources, knowledge available from the scientific literature, from different databases and other knowledge sources to try to help support interpretation from genomes to phenomes. So this include, again, literature report, network analysis, variant analysis, as we heard again from previous discussion about the variations in the genes, the proteins can really lead to vastly different protein functions, subcellular localization, and disease consequences. And so all those type of information, and certainly drug target identification. So again, a critical aspect for enabling effective disease treatment is understanding the mechanisms, the effect of variants have on protein function, and therefore to help disease understanding through proteins. And so much of the functional information in UniProt are knowledge gleaned from the scientific literature. So our group, along with our collaborators, has been engaged in natural language processing research for quite a number of years to mine the scientific literature. So we have developed a workflow, a text mining workflow that will allow us to mine from the scientific literature, from the abstracts, from full-length articles, to extract the entities. So these are the entities, basic entities like genes, proteins, but also very importantly, composite entities, which variants, what mutations on which genes, what post-translational modifications on particular proteins and particular residues. And not just entities, but also extract information regarding the relationships among those entities and the biological context where those genes and entities exist. And in order for us to integrate later on then the knowledge gleaned from the scientific literature to other data sets, to large scale data being generated, to, again, knowledge bases, we need to provide the mapping that we call the normalization, where we can map ID, the name, the sequence, to databases, to ontologies and control vocabulary, because that's then when all the different entities relations could be connected in the semantic environment for us to do more interpretation. And so that's the semantic integration, where data from the literature mining get to integrate it with data from disparate resources. And then this could be represented, visualized for analytics to facilitate hypothesis generation and knowledge discovery. So for the information retrieval and relation extraction, we have used a so-called a generalizable framework, the so-called extended dependency graph, EDG. So the idea is that with this kind of generalizable framework, we can leverage syntactic variations and automatically generate extraction patterns. So with the generalizable framework, then we can adapt the system to quickly develop text mining tools tailored for different bioentities, different relations of interest for different problems at hand. So with the advancement, again, of the data, the deep learning models, as many have previously discussed, such as the PCNN, the pulse coupled neural network model, and the bidirectional LSTM, long short-term memory model, they have really led to major improvement in natural language processing system performance as well. But this would require, again, large training set, manually curated training set for the new tasks. So what we have done is to develop distant supervision to augment the training set with automatically obtained data sets, and then also develop transfer learning to improve the performance of both distant super learning data and the human label data. So with this kind of framework and the enhancement, we have developed a good number of text mining tools that allow us to extract entities and relations from scientific literature for us to build knowledge networks, biological networks. So for example, we have the RLMS-P that allow us to extract information regarding kinases, their substrate, the phosphorylation sites. and allowance PTM for other PTM enzymes and other substrates. The EFIP is a program that allows us to look into the interactions of phosphorylated proteins. So again, we are very interested in protein-protein interactions, not just at the gene level, but really at the modified protein level, because different interaction partners of different modified forms really lead to different biological processes and biological function. And it's really important to capture all those different kind of forms. MicroRNA gene regulation, information, disease to mutation association, role of microRNA in diseases, gene protein name detection normalization in plants, and eGuard for studying genomic anomalies and drug responses. So bring the gene, drug, disease variants information together. So a number of text mining tools were developed. And so in the context of big data and precision medicine, it then allows us to look into the genetic variants, whether they are disease-driving, disease-causing, or disease-associated protein entities. We will be able to look at them at the very precise proteoforms levels, whether we are talking about isoforms, variants, talking about modified forms, post-translation modifications, or protein complexes. And so it provides then a very rich entity relationship for different kind of biological context. And again, through the full-scale processing of the PubMed abstracts and the PMC full-length open access data set, it allows us to construct the knowledge networks. So we have developed with so many disparate text mining tools that we have developed over the years, and also a number of tools in the public domain, we have developed this automatic workflow where we can integrate multiple text mining tools for us to do semantic text annotation on large-scale text. And so start from the input text, we have all the different text mining tools Dockerized. The texts are aligned so that we can extract the entities, relations, and do the normalization. And then all the information is kept in the database and disseminated in multiple format. The output format in BioC, which is a standard format for other text mining tools, in XML, in RDF, the RESTful APIs for computational access of the semantic text annotation results. And again, everything is Dockerized, so it could be reused. And so essentially, it's a FAIR principle: findable, accessible, interpretable, and reusable for others who are interested in building their text annotation system. So this is just an example of when we start building a framework, a network like this, a workflow like this, it allows us to then gather information from across documents to already construct some meaningful gene disease drug network. So this is an example using SEP-B1 as a query term. And so we use the SEP-B1 to extract all the scientific documents that contain information regarding those entities in our that we have captured in our text mining tools. So the take-home message here is that when you start combining knowledge across multiple documents, you can look into the function, you can look into the regulation in disease, you can look into the drug response. And so I don't have to go through the detailed findings through those documents, but a take-home here is that, for example, one can find the controlling regulators of expression of SEP-B1 could be a potential point of intervention to overcome drug resistance because we, from this network, we know how they are regulated by different microRNAs, and we know how the phosphorylation network impact the drug response. So that's kind of the first part about this literature mining and knowledge integration. I wanted to then go into the semantic computing and knowledge discovery aspect. So talk about the link open data and how we build, again, this kind of semantic network. So with this, we really expand on the work that we have done on protein ontology that capture knowledge about proteins to model biology. So essentially, we wanted to connect the various protein types and the relationships. So for this, we have the ontology to capture information about protein classes, the evolutionary relatedness of whole proteins, like what was discussed earlier. And ontology for proteoforms. So that's the forms derived from genetic variation, alternate splicing, proteolytic cleavage, post-translational modification, and so forth, protein complexes, and protein sites. These are the individual amino acid sites that have important implications, whether we are talking about catalytic residues, whether we are talking about epitopes, ligand binding sites, drug binding sites, protein interaction sites, other structural functional sites. So then we have with the information with the protein ontology as the foundation, we have developed and presented the protein ontology using the protein ontology linked open data framework to help support semantic data integration and computing. So we use the RDF format and disseminate via the Sparkle endpoint. And the protein ontology link open data allow us to then expose, to share, to connect data and knowledge about protein-related entities. So currently, the protein ontology has nearly 12 million triples, and it is served by an open link virtuoso server along with metadata description in the W3C HCLS specification. It's also one of the ontologies in the BioPortal repository. Indeed, it is one of the largest, the fifth largest among nearly 850 biomedical ontologies in the BioPortal. And here it's just an example of a RDF data model for a particular phosphorylated protein. So with the RDF model, we can then readily generate knowledge graph computationally by using federated Sparkle query that integrates data from multiple data sources and annotation types. So the figure here shows that we generate a model using the protein ontology RDF, the UniProt RDF, and also the RDF of this resource called DisGenNet that provide rich information regarding gene disease information, disease drug information. And so together, this provide rich and traceable annotation on Alzheimer's disease protein variants, and along with the confidence scoring. And for this, the information from the underlying sources also get leveraged, such as the Arts Forum, that provide a lot of very rich annotation about Alzheimer's proteins in protein ontology. So then go one step further with the RDF model. They then become the building blocks for us to generate knowledge networks. So we can integrate results from text mining, from data mining, from semantic queries, to build knowledge network. And again, this is just as an illustrative example. The example here shows a genotype-phenotype drug network for 6 genes that are implicating Alzheimer's disease. So the information include gene variant disease relation obtained by text mining using the eGuard text mining tool. Also information data mining from resources like ClinVar, and the gene drug therapeutic relationships obtained by Sparkle query of drug bank. So again, it shows the relationship between the gene drug disease and how they are connected. So as we all know, with the COVID-19 pandemic, the research community is also fully engaged in scientific endeavors to address the pressing need in COVID disease understanding and drug discovery. So So the major consortium effort has led to rapid release of COVID-19 open research data sets, the CORD-19, that consist of scholarly articles about novel coronavirus from Medline PMC, bioRxiv, and other literature resources. There's also the LitCove curated literature hub for information on SARS-CoV-2 and the disease, and as a central access to relevant articles in PubMed. So with the availability of this massive literature information, we also see text annotation effort on COVID-19. So for example, the NCI PubTator text annotation has covered hundreds of thousands of papers and millions of entities covering species, disease, gene, chemical, cell line, mutation, and so forth. So with the information that can be integrated from text mining, data mining, and using Sparkle queries, we try to build a knowledge network for drug repurposing use case for COVID-19. So again, the relationships and the different entities are retrieved from the different resources. So I don't think I'm going to go over with the details as to where and how this information were extracted. I just wanted to show you this resulting network, the knowledge network, centered in the ACE2, which is shown in red, the red node, and the interaction partners in pink, as well as the COVID and comorbidity diseases in blue, and the gene variants in gray, and therapeutic drugs in green. And so what is particularly interesting here is that the drugs with dark borders. So again, the therapeutic drugs are those indicated in green. The ones in dark border are those drugs associated with COVID-19 in the literature and in the clinical trial in therapeutic information browser hosted by the COVID-19 healthcare coalition. So one can go one step further. I know this is an extremely busy slide, but instead of focus on ACE2, which is a very important, basically the cellular receptor that binds to the viral protein, the spike glycoprotein that constitute the entry point to the SARS-CoV-2 into the human host. We can take the top genes from the literature that implicate in COVID-19 and then build this kind of knowledge graph and identify therapeutic drug targets that associated with different steps involved in the COVID-19 infection. And so even though this is a very busy network, the take home again is that we can then identify more relevant drug targets, including many more are currently under clinical trial. So, so collectively, I think literature mining, data mining, semantic computing, and knowledge integration are really integral to the development of what this workshop is about, the knowledge guided machine learning framework. And so with the advances in high throughput experimental biology from genomics, epigenomics, transcriptomics, proteomics, metabolomics to microscopy, biomedical imaging, flow cytometry, and so forth, and also digital transformation of biomedicine with wearables, with sensors, the electronic health record, the GIS mapping in public health and epidemiology. Certainly there is no shortage of big data. So they really would create unprecedented opportunity for us to do knowledge-guided discovery in biology, in precision medicine, in digital agriculture. So to harness the data revolution and accelerate research in data science certainly requires interdisciplinary research collaborations across fields impacting society so well put together by this workshop. So at University of Delaware, we established the Data Science Institute to bring together faculty and students from all 8 colleges across campus, from health sciences, physical sciences, environmental sciences to behavior, and social sciences and public policy. So we would be extremely delighted to really contribute to the development of the foundations and applications of the knowledge-guided machine learning. And I think the KGML would be a really ideal framework for collaboration, for translational biology and for team science, bring together us at the interface of biomedical and computational sciences. So I just like to end with an acknowledgement to the amazing team that we have at University of Delaware, at Georgetown University, and our partner institutions, and certainly our funding agencies. And very special thanks to organizers for this very inspiring knowledge-guided machine learning workshop. We are very much looking forward to a close collaboration with this broad community. Thank you.
Thanks very much, Cathy. We're right to time. And one thing, I do want to have, we have one question on the Slido that I'll get to, but I don't want to forget to mention that Cathy has kindly agreed to be one of our panelists this afternoon. So if there are additional questions, they can be directed at that time. The one that I see here on Slido is I think a straightforward question about how conflicts in the literature are resolved. You bring together all these different sources and there are controversies in science.
Yes.
Is that handled algorithmically? Does that throw an error? How is that handled?
Yeah, so so what we have been doing is to provide confidence scoring. So depending on the underlying sources of the knowledge, if we have more sources that providing consistent knowledge, then they could be getting high scoring, whereas the others, because oftentimes they are even conflictory information, and so those are highlighted. So one way is to provide evidence attribution, another way is to provide the scoring factor, and also a lot of the manual curation go into places for knowledge bases like UniProt. And so that provide excellent ways for us to oftentimes resolve this kind of conflicts as well.
Excellent. Well, I'd like to thank all of our speakers from the first half of this session, and thank you for all of the attendees for sticking with us. We will now take a 13-minute break and reconvene promptly at 11:10. See you then.